Hello everyone, welcome. This is Sanjeevani Nayak. Now in this video, I will be doing diagrams, all the diagrams which can be asked for drawing in ICSE Biology syllabus from Unit 1, and you should watch this video completely so that you don't make any mistake while drawing diagram. Let us begin. Now I'll start with chapter 2, chapter 1, that is cell. It is for revision only and no diagrams are asked from that particular chapter. Second chapter, that is structure of chromosome, cell cycle and cell division. Cell division, all the different phases of mitosis can be asked in diagram, but we will start with structure of chromosome. Now, the first diagram that can be asked to draw, and these diagrams which I will be explaining can also come for identification. Okay. And labeling. So if you watch this video, you will not only learn how to draw the diagram, what common mistakes we make, how to avoid these mistakes, but also you will be able to understand the labeling and how to identify the diagrams. Okay, so let us begin. So the first diagram which comes in structure of chromosome, which can be asked for drawing purpose, is the structure of nucleotide. Now this can be asked for labeling as well. Okay, now the structure of nucleotide, if you remember, nucleotide has three components. Yes, pentose sugar, nitrogenous base and phosphate. And this is what we have to draw. Now pentose sugar, we are going to draw pentagon, then phosphate will be represented in circle and nitrogenous base in the form of rectangle. So I'll show you how. Now when you are drawing, there is no need of scale. You can do freehand drawing. Only thing is it has to be neat and well labeled. So first we will be drawing pentagon. Okay, then one rectangle attached to this pentagon and then one circle. Okay, now labeling. This is pentose sugar. Phosphate, and this is nitrogen base. Now student, it is very important to give the heading to the diagram. So below the diagram you must write the name of the diagram. Now second question is duplicated chromosome. Now very often it is asked, duplicated chromosome. Now when you are drawing duplicated chromosome, you have to keep in mind, duplicated means क्रोमैटिड का ऑलरेडी कॉपी फॉर्म हो चुका है, मतलब there will be two sister chromatids in duplicated chromosome. We'll label this as centromere and this two as chromatid. Done. Okay. Only these two labeling has to be done. Now कभी-कभी ऐसा हो सकता है कि आपको यहाँ पर sister chromatid लिखने बोले, so that is okay. If you write sister chromatid or chromatid, both are fine. So this is duplicated chromosome. अब single खाली chromosome बोला तो you can even draw the single one. Now if you want to draw the single one, it will have only one chromatid, one centromere and one chromatid. Okay, so you label this as centromere and this as chromatid. That is enough. Okay, this is also chromosome and this is also chromosome, but this one is duplicated chromosome. Okay. Now next, the diagram which you have to draw, which can be asked for drawing, is crossing over. When you draw a diagram for crossing over, there will be two diagrams. Okay. That is during crossing over and after crossing. This is when crossing over take place. I will show this chromosome shaded. And this is chiasma or crossing over. After crossing over, we will show some part as colored over here, some part uncolored. So what you have to keep in mind in this diagram is, if you are showing paternal chromosome as colored or shaded, so after crossing over occurs, after chromatid exchange occurs, you will show some part of paternal chromosome in maternal chromosome and maternal chromosome in paternal one. So this is the next. Let us see the cell division, that is mitosis. In animal cell and plant cell, I will draw simultaneously. Now we know that mitosis has two phases, right? One is karyokinesis and the second one is cytokinesis. Okay, and we know that this karyokinesis is further divided into four phases, that is PMAT. Okay, so prophase, metaphase, Anaphase and Telophase. So all these phases we will see. First I will draw for prophase and metaphase. Now what you have to remember is, how many chromosomes should we draw? Now the chromosome number, if it is given in the diagram, that draw neat and label diagram of animal cell in prophase having 4 chromosomes. So you will draw 4 chromosomes. If it is not mentioned, then you will draw as per your wish. Okay, I will be showing you with 3 chromosomes. If the question is that, if from the diagram based question, like if a plant cell or animal cell in prophase is shown and they ask you to draw the next phase, then you will check the number of chromosome in prophase and using same number of chromosome you will draw the next phase, that is metaphase, or if the question is previous phase, same. So number of chromosome, how many chromosome you have to draw, that completely depends upon the question. If nothing is mentioned, you can draw as per. If it is independent question, then you can draw as per your wish. Okay, so let us see prophase. First I will draw the diagram and then I will do the explanation. Now in this diagram, remember prophase, you don't have to draw early or you don't have to mention early or late phase. Like for all the phases, I am talking about all phases. So you just have to make prophase. Now whenever we are making prophase, one thing we have to keep in mind is nucleus. We should show nucleus in dotted with dotted line. Now that dotted line shows disappearing nucleus. Okay, so nucleus should be disappearing and you can see chromosomes are randomly arranged. I have drawn 3 chromosomes and then of course we will label it. So let us label. Okay, now you can see animal cell. So the outermost is covering a cell membrane. Then you have to show the dotted circle which you do as disappearing nucleus. Then you can see centromere. Now you know centromere, the constriction in chromosome. So that you will label as centromere. This will help you to label also if the diagram is given. Then you can see the arm of the chromosome I have mentioned as chromatid. Then now this ester, centriole and centrosome. Now what is this? I will explain you. Then you can see the line which is going between the two centrosome is called as spindle fiber, or the thread-like structure which attaches to the chromosome, that will be labeled as spindle fiber. Now ester, centriole and centrosome. Now this labeling is very important to understand. Sometimes I have seen that students lose their marks in this. Now when you are drawing centrosome, so see these two, one dot and one line, these two represent centriole. So if the diagram has, like, if label is here, if the label is here, this will be centriole. Okay. Now when to write ester? Now we know that from this star-like structure comes out. Now this, if the diagram shows this, then you will write ester. Okay. And this both together is called as centrosome. Okay. Both is together called centrosome. So this is what you have to keep in mind. Now it's okay if you don't write centrosome, or if you only write centriole or centrosome, that is okay. Ester nahi likha to bhi chalega, but right now I have included in the diagram so that you know what are the different parts. If this comes for labeling, you should not miss it. Now I will draw the plant cell. Now there are two differences in animal cell and plant cell. One, cell wall is present, and second, centrioles or centrosomes are absent. So when you draw, सब कुछ वैसा ही रहेगा. Only thing is, you are going to draw one extra wall, that is cell wall, okay? And then dotted disappearing nucleus you will draw. Then spindle fiber comes somewhere from here. It's okay if you don't draw all spindle fiber in prophase. Okay, many spindle fiber in prophase. And then chromosome. Okay, so this is the difference. Baki labeling is same. This will be spindle fiber. Then this is the disappearing nucleus. Then this is the cell membrane. Extra you will add cell wall. Okay. Then centromere here, you will label it differently. And then this is chromatid. बाकी तो सब सेम रहेगा. Okay, सिर्फ क्या क्या ऐड हो रहा है यहाँ पर, cell wall ऐड हो रहा है and centrosome is missing. So this is all about the prophase, that is first phase. I hope it is clear. Okay, now itna sara labeling will not do in the metaphase. Okay, we'll see what important labeling has to be done. So next is metaphase. Now when you are drawing metaphase, you have to remember that there is no nucleus. So nucleus completely disappears and chromosomes are aligned in the equatorial plane or central plane. So you will draw 3 chromosomes. Now since I have drawn 3 chromosomes in the upper phase, that is prophase, you will also draw 3 chromosomes aligned at central plane. Okay, I will draw first animal cell. And then same centriole. These are the 2 centrioles, aster and spindle fiber. Now spindle fiber is attached. Spindle fiber is attached to centromere, chromosome at centromere. So this is what you have to draw. Of course you can draw few more extra, nahi kiya to bhi chalega. Okay. And then in plant cell same, but here there is no point of origin. Centriole nahi hai, centrosome. So this is how you will draw metaphase. Labeling same. Only thing is here you will add one more extra covering, that is cell wall. So this is cell membrane. Okay. Then here this is spindle fiber. This is centromere. Then this is chromatid. In plant cell, you will add one more thing, that is cell wall. And in animal cell, you will label this entire thing, you will label as centrosome. It's okay if you don't label aster and centriole. See the centrosome label here? That is fine. Okay. So these labelings are important. Now let us see anaphase. Now anaphase, remember, you will not show constriction in cell membrane of animal cell in anaphase. It will be seen only in telophase. So when you are drawing, now there were 3 chromosomes, and these 3 chromosomes in anaphase, it splits, that it is moved away. It moves away from the equatorial plane. It breaks, that is sister chromatids move apart from each other. It breaks at centromere. So you are going to draw that. And again there is no early phase and late phase. Now this either you will write chromatid or you can even write daughter chromatid, but don't write sister chromatid, since it is sister chromatid is separated. You cannot write again it as sister chromatid. You will write daughter chromatid or simply chromatid. And then this is centrosome. That is enough. Okay, and if you want to mention centromere, here we can mention centromere. Now in plant cell, cell membrane, spindle fiber, centromere, this point is centromere, this is chromatid. You will add one more, that is cell wall. Okay, rest is same. Okay, so this is anaphase. Remember, when you are drawing, you will not show constriction in anaphase of animal cell. So this is animal cell and this is plant cell. And then comes telophase. Now what is important in telophase? Showing reappearing nucleus. So you show nucleus which reappears in dotted form. And these chromatid which you have drawn over here, it decondenses. It decondenses, that has become thread. Abhi, each cell will have 3 chromatid, right? So you will draw 3 thread-like structure. Don't draw more. 3 thread-like structure, and these 3 thread-like structures are the chromatid. This is ester. Again, spindle fiber is disappearing, so no need to draw more. And then there is nucleolus which reappears. So you can mention. So labeling quickly. Nucleolus, then chromatin, then centrosome, then here, this is important to mention, reappearing nucleus. Okay, so this is done, and then here this is furrow formation. Done. Here there is no furrow formation, so no need to show furrow, but you will show the central plate. So show central plate, reappearing nucleus and chromatin. So label this chromatin and of course nucleolus. So nucleolus, this is reappearing nucleus, this is cell membrane, add this as cell wall, and this is central plate. Central plate. And now cytokinesis. In cytokinesis, in animal, you will draw 2 cells. 1, 2, and both with proper nucleus. Proper nucleus, nucleolus, and so here cytokinesis has occurred. And of course, 3 chromatin. Now, cytokinesis is not very important. Nucleolus. This is the cell, outer wall is the cell wall. Then this is cell membrane as we have done before. Okay. Then nucleus and nucleolus, etc. Now these cells are called as daughter cells. Okay. And here also daughter cells. So I hope you understood. Now whenever you are drawing telophase, in telophase only you will draw furrow formation. Now sometime in diagram they show furrow formation in anaphase. So when you are labeling, you will label not on the basis of furrow formation but on the basis of chromatid. If chromatids are separated, sister chromatids are separated, then you will label it as anaphase, then that diagram is anaphase. And if you see reappearing nucleus, then you will label it as telophase. So this is done. So plant cell, animal cell, cell division, that is mitosis, I have completed. In Unit 1, the chapter which is important as per diagram is the second chapter, that is structure of chromosome, cell cycle and cell division. If you have any doubt, mention in the comment, and don't forget to subscribe and hit the bell icon. In next video, I will be doing list of diagram from Unit 2, that is Plant Physiology. Thank you very much.